Hello students, this is Dr. Anjani, Assistant Professor in Biotechnology. Welcome to e-learning sessions in Medical Biotechnology. Today in this session, we learn how enzymes are used as therapeutic agents. The uses of enzymes as diagnostic tools, as prognosis tools, and as disease markers is known to medicine for quite some time. More recently, enzymes have found the applications as new class of therapeutic agents. This new emerging field is named as enzymotherapy and will provide new class of biological drugs during the next decade. More than 200 enzyme deficiencies related to genetic dysfunction or genetic disorders have been reported in humans. Research in enzyme therapy and gene replacement therapy is currently in progress throughout the world. But the enzymes have a specific characteristic to have wide applications in therapeutic treatment. These are used as therapeutic agents because they are used in therapeutic or replacement therapy in inherited genetic diseases. These are used as scavengers to limit the tissue damage during the course of inflammatory diseases and in various ischemia. These are used as specific detoxification agents during acute chemical poisoning. These are used as temporary palliative agents during an acute organ failure, and they are used as anti-neoplastic agents. Though they have all these characteristics to consider in therapeutic applications or in therapy, they have some problems like it's very difficult to clear the enzymes. Rapid clearance of the enzyme from the blood is very difficult, and they elicit the immune reaction because they are proteinaceous. Avoiding immunogenicity is very difficult, and when we are using them in the therapy, it is we are using in their high pure form, having low Km and high Vmax, to use them in very low concentration. That is very difficult and cost effective, and the enzyme has to retain its activity under various operational conditions. But we know the enzymes, they have the optimum conditions in which they exhibit the activity. Let's see few enzymes and their therapeutic applications. Streptokinase and urokinase, they are used to cure the clots in the brain. Trypsin, lipase and amylases, they are used in pancreatic insufficiency. Asperginase and glutaminase, they are used in acute lymphoblastic leukemia. Hyaluronidase is used as to increase the anesthetic behavior, and they are used for easy diffusion of fluids. Papain is used as anti-inflammatory drug. Chymotrypsin is used as a painkiller and as anti-inflammatory agent. Alpha-1 antitrypsin is used in deficiency and emphysema. So like this, you have many enzymes having the therapeutic applications. Here are a few enzymes, and we see their therapeutic applications in detail. So here DNase should be written like D and N should be capital, and ASE should be in small letter. So it's a typing error. DNase, it is used in curing cystic fibrosis. Cystic fibrosis is the genetic disease. Excessive salt loss occurs in the sweat of the people suffering from the cystic fibrosis, due to which this excessive loss of salt is due to malfunction in the ion transport, It produces extremely viscous mucus in the respiratory tract, which is very optimum for the establishment of the bacterial infections. Neutrophils are attracted at the sites. Neutrophils causes digestion of the bacteria. DNA from the bacteria is released and it interacts with a variety of extracellular substances present in the infected lungs. This results in viscous mucus. DNase, which is used as aerosol, can reduce the mucus viscosity. Human DNase is now produced in relatively large quantities. Clinical trials are under process. Next we'll move on to the debriding enzymes. These enzymes effectively clean open wounds by removal of the foreign matter and any surrounding dead tissue. This allows rapid healing of the wound. These enzymes are applied only topically to the affected areas. Debriding enzymes example includes trypsin, papain, collagenase. In digestive aids also we use some enzymes like alpha amylase, lactase, papain, pepsin. These are useful as digestive aids and they carry out breakdown of number of dietary components. In the pancreatic secretions usually contains lipases which digest the fats, proteases which digest the proteins, and amylases digest the starch or the carbohydrates. Supplemental pancreatic enzymes are found in pancreatic products to get highest potency. Next we will move on to the therapeutic application of asparginase. Asparginase catalyzes the hydrolysis of the amino acid aspargine and yields aspartic acid and ammonia. This enzyme can be purified from variety of microorganisms including yeast, fungi, and even from bacteria like E. coli. All cells, normal cells, require aspargine to sustain the normal metabolic activity. Most of the cells, they have the capable of synthesizing this amino acid except certain cancerous or the malignant cells. The administration of asparginase thus reduces or prevent the growth of such cancerous cells by reducing the level of aspargine availability to them. Normal cells do not have such a deleterious effect. Asparginase enzyme preparation used for therapy is normally purified from the bacteria, that is E. coli. E. coli produces two forms of asparginase isoenzymes, of which only one is clinically effective. This enzyme is used in acute lymphoblastic leukemia and acute myeloblastic leukemia in children and adults. Next we will move on to the superoxide dismutase therapeutic application. This is the protective enzyme which will convert the superoxide radicals and hydrogen peroxide produced during the metabolic reaction to water and oxygen. So here the two protective enzymes are forms of superoxide dismutase are found in eukaryotic cells. This superoxide radical convert this superoxide radicals which are highly reactive into H2O2. Then this H2O2 will be converted into water by catalase enzyme. Human superoxide dismutase, that is HSOD, may be produced by recombinant system. Such preparations are under clinical trials. Renin, it is used to control blood pressure. Streptokinase and urokinase, as I told earlier, they are used to remove the blood clots. Streptokinase is it is the microbial origin enzyme used in therapy. Streptokinase is produced by Streptococcus species, can break down the blood clots in veins and prevent heart attacks and strokes. A streptokinase enzyme is very promising drug, but because of its microbial origin may provoke or elicit immunological reaction. Urokinase is enzyme of human origin and has similar properties as that of streptokinase. This urokinase is extracted either from the urine successfully or it is obtained from the kidney cells, but very soon the relevance gene which is responsible for your production of urokinase is introduced in bacteria by genetic engineering to produce this enzyme, but it is very cost effective to produce in plenty in market. Lipases are very important in avoiding Gaucher's disease. It's one of the known hereditary disease due to lipid storage. Children suffering from this disease lack enzyme which normally break down lipids in the body, that is glucocerebroside. This material then accumulates in the lipid spleen, bones, producing swellings, causing damage to the nerves and death of children before the age of 2. If the right enzyme is injected into the cells, the child could be relieved from the symptoms of Gaucher's disease. At present, enzyme is purified from the human urine or placenta, where it is present in very small quantity. The major disadvantage is its cost. Genetic engineering has made it possible to produce this human enzyme in bacteria, but for active enzyme to use in the therapy, it requires post-transcriptional modification, that is glycosylation. Next is adenosine deaminase. Deficiency of adenosine deaminase, a cytoplasmic enzyme found in high concentration in lymphoid cells, cause combined immunodeficiency disease due to lymphocytotoxic effects of adenosine and deoxyadenosine. So this deoxyadenosine causes DATP pool, deoxy condition. ATP pool will be therein, so that is deoxy condition, which blocks the DNA replication by inhibiting ribonucleotide reductase. Enzyme replacement therapy in adenosine deaminase deficiency disease is approved by FDA, that is US Food and Drug Administration. So in this session we learned how these enzymes are used in therapy to cure the diseases. I hope you understood this session. Thank you.